Good morning, everybody. This is our next global online biodiversity informatics seminar. In fact, it's the 9th in the series. Just looking forward a bit, I'm afraid that the last Thursday of the month is Christmas Day, and so I think we will take a month off, and so we will have a next seminar in late January. I'll be making announcements about that. So we got a month off. For the moment, however, we have Andres Lira giving today's presentation. Andres recently completed a Ph.D. here at the University of Kansas and is now moving back to home in Mexico and starting a researcher position at the Ecology Institute in Jalapa, Veracruz. And today's seminar will be a bit of a view of what he'll be doing in that position. So this will be new material both for me and for you. So thanks, Andres, for accepting the invitation, and it'll be good to hear what you have to say. Anybody who has questions about Andres' seminar, please be certain to send them to biodivtraining@gmail.com and get us as many questions as you can as soon as you can, because otherwise it will be hard to get those questions to Andres while he's still broadcasting. So, Andres, once again, thanks for your morning, and all yours.
Well, thank you, Dan. It's a pleasure to be here with you all. Happy Thanksgiving for those that celebrate it, and I'll try to tell you the story with something that I'm involved right now in my new job in Mexico. I'll be talking about how we are thinking on exploring potential invasion of forest pathogens. This is something that has to do with, first of all, crops in Mexico, mainly avocado. It's a economically important crop in Mexico. About 85% of the... Can you see my screen? Okay. There we are. And so I was saying that this is a project that started with potential pests for avocado, and now we see it's more than avocado. It's more about not only that important, economically important crop, but also other forest ecosystems. And we are using ecological niche modeling and other ways to measure or assess biodiversity related mainly to the beetles that attack the forests. These are beetles that come from Asia and are exotic in the Americas, and that's the reason we have it's of big concern, because they can transmit the fungi that are in symbiosis with the beetles, and once they get in contact with native beetles here or the forests, this may become something that can be drastic for forest ecosystems in general. This has been a collaborative effort started with the Institute of Ecology in Jalapa, where I will be working. I'm a researcher there right now. And people from CONACyT, the National Commission on Biodiversity from Mexico, the major biodiversity database from Mexico is there, and tools for doing remote sensing and other habitat quality layers that we will be showing today come from CONACyT. And then important is the participation of governmental agencies like SAGARPA, and they have provided us with occurrence data of the pests, and finally the collaboration with my KU former advisor, Jorge Soberon, with whom I've been sharing this analysis with. This seminar I'm presenting today has an inspiration, or has been inspired in previous workshops in Mexico related particularly to ambrosia beetles and avocado trees, avocado plantations. This is mainly in the region of Michoacán. But in general, as I mentioned at the beginning, this is of a broader importance. These technical workshops, or workshops on the ambrosia beetles that I will be talking about today, have been a big inspiration, as I mentioned, because I've seen the link between academia and people that is directly working in the field, basically field technicians that work directly with these crops. And they see the world in a very different way than we do. No? I'm comparing academia and producers, basically. And so that exchange and the need to be able to talk to them and to have not really a solution, but some means to assess the risk of these beetles in these ecosystems is very important. And their information is very valuable for us to improve the models that we're presenting, and I will be presenting here. So the problem, in general, we can think of it like that we want to understand the diversity patterns first at the meso and macro scales of hosts and vectors of these forest ecosystems. So we want to predict the potential distribution of hosts and vectors at geographic ranges and their ecological niches. And the basic question we are after is, what are those requirements ecologically and geographically? How far can they get? And how could they move around in the landscape in order to reach, as I mentioned, both economically important crops and in general other forest ecosystems? So those are the main problems and questions we're facing. For me, it was of great inspiration to read this paper and related papers, but basically this paper that is first author Greg Gilbert, that was here at the University of Kansas, where he explores the relationship of phylogenetic diversity, as you can see on the lower right panel. So how closely related are species on a phylogeny, in this case hosts, and their probability to be infected by a pest. And a general pattern is that the more closely related they are, the likelihood of being infected increases. So in this panel in the middle, you can see there is a phylogeny, a host phylogeny. You can see different ways of arranging the ranges of species, but looking at it through the phylogeny. So you can see there is a narrow range on black dots, for example. So this is a phylogenetically constrained host range. You can have another, like this is case A. In case B, for example, you have what we call a narrow range, but it's a phylogenetically dispersed. So these are few hosts that can get infected, but the way they are clustered in the phylogeny, or how closely related they are, is very different. Or the third case would be when you have a wide range of hosts, and this is you have many more hosts that can get infected, so a more generalist pest is affecting these hosts. So this is important because what we have been doing here for my dissertation, and my approach is very geographically oriented and thinking about these problems in terms of phylogenies and how closely related are the species that can get infected is important, and having this combination of or perspectives of biodiversity in hand, I think, is very important. So this is just an inspiration for me and something that is in process, not yet completed, but we are starting to think more on these terms. So for today's seminar, I will first introduce two key concepts that I will use. The first of them is range diversity plots and ecological niche. You may be more familiar with the second, and I will spend a little bit of time in each of those. Then I'll introduce which are the two species of ambrosia beetles that of big concern for us in Mexico, as well as in the US, where they have been introduced since around 2002 and 2003 in both coasts, East and West, Georgia and California, respectively. And then I'll show you, using GBIF information, what the Lauraceae species diversity patterns look like. The reason of using Lauraceae species is because one of these beetles is particularly affects this family of plants. And then I'll concentrate in how this plant family looks like in Mexico, how it is, where are the hotspots of diversity of this plant family. Then I will try to combine all this information on spatial diversity of beetles and plants to understand the potential risk of invasion of this particularly one beetle species, Xyloborus glabratus, using spatial multicriteria evaluation. Finally, I'll show some of the potential ranges of plant species that have been affected in California, that are also in Mexico, and where the second beetle species, Wallacea, can be introducing in Mexico, can be spreading through to Mexico through the border. No? So the first important concept to talk about is range diversity plots, and we depart from what we call a presence-absence matrix. So you can see on the right panel, you have the summary of species diversity in sites. So if you divide geography on whatever grid you want to use, let's say you are dividing your landscape or your continent using a grid of whatever resolution you want, and you have each of those cells is here represented by the sites. So you have, in this case, 3 sites, and you summarize how many species are there. No? So you have, in this case, 4 species. So you can see you have 2 very basic measures of diversity. If you sum the columns, you have what would correspond to the range of the species. So there is, in this case, the first species is very widespread. It's occupying all the sites in the landscape. And there are, in other cases, the other 3 cases, these are very narrowly distributed species. They are only occupying 1 site. If you look at the sum of the rows, you see the richness of each site. So again, you have sites that have many more species compared to the others, right? So you have these 2 very basic measures of diversity. This presence-absence matrix, then we can extract from it very important measures, like I mentioned, alpha diversity, which is the local species richness, or the turnover of species, beta diversity, how widespread are the ranges of the species, which would be the species range size, and all the other measures of ecology that we have been using through time. No? However, there is something else that we can extract from the presence-absence matrix that can be richer. No? That something where we combine richness and range in one graph, and that's what we call the range diversity plots. So this is what I'm showing up right now. If we extract the proportional richness of species and per site, and the average proportional range of the species that inhabit one cell, what we call the dispersion field, and we plot them, we get this characteristic plot that we see here. And I think Jorge Soberon has already spoken about this in a previous seminar. So you have on the x-axis what we call the mean proportional range size, So this is, on average, how widespread are the species that inhabit each of these cells. So you see each of these green dots corresponds to one site in the geography. On the y-axis, you have what would correspond to the proportional species richness. So on average, how many species are there in each cell? So you can see this is a little bit more interesting than just having an idea of how many species or how widespread they are as a sum of the presence-absence matrix. But if we combine them in this plot, you can immediately detect, for example, if you go to the lower right of the plot, you can see these sites here on the extreme right correspond to sites that on average have species that are very widespread. If you move to the left, you can see that these sites on average contain species that are very narrowly distributed. And if we move up on the plot, you see sites that are in general very narrowly distributed, but are very rich in species. So you can see the use of this, we'll see that later. I mean, applying it to the Lauraceae species and the Xyloborus or the ambrosia beetles that I'm talking about. For us, it's also very important not to only look at this view using sites, but also the species. So if you transpose the matrix, instead of having it as we had it before in the upper panel, we have it as it was before, sites and species. If you transpose this matrix, you now have the view per species. So you can see, for example, on average, how diverse is the community of species that each species shares its range with. So this is what we're representing it here. Now, I use the same plot. I apologize for not changing the plot, but the idea is the same and the shape would be approximately the same. The number of points on the plot would change according to how many sites or species you have. So in this case, I'm using the same plot per sites that I had before, but notice that the names of the axes change, right? So now the y-axis has a proportional range of the species, and on the x-axis you have the mean proportional species diversity. So as I mentioned before, now this x-axis is telling us on average how diverse is the community of species that a species shares its range with, and on the y-axis again how widespread or narrowly distributed those ranges of those species are. I'll be showing this applied to our system here. Now the second concept I want to mention is, because we're using it a lot for what we're showing here, is the ecological niche concept and its relation to geographic space. So we have, and this cartoon, well, not really, it's actual real data on mammal Antilocapra americana, the pronghorn. What you see on the left panel is the three-dimensional niche space of the North America, or I would say the environmental combinations available in North America are in black, and then in the yellow ellipsoid that is here is what would correspond to this species niche, right? So if we draw an ellipsoid using these three dimensions, precipitation, temperature, maximum temperature, and minimum temperature, we can draw this ellipsoid, and if we fix it, and we see the niche, right? And then we're asking, well, we're not asking, we're moving the time, so the environmental combinations are moving from 135,000 years ago to the present. So that's why you see this cloud of points moving. And you can see, so this is basically the last interglacial here, then goes to the last glacial maximum, and again to the present. So the last interglacial and the present look very similar. And when you see the cloud of points very clustered, it would correspond to the last glacial maximum. But anyway, then I know by now you probably have noticed how the yellow dots are changing in the geography, which is in the right panel. So there is a correspondence between the environmental space that you see on the left panel and the geographic space. So where are these environmental combinations? And as I mentioned before, yellow dots correspond to the environmental combinations that correspond to the niche of the species. So this is basically the idea. We're using environmental predictors to try to predict, where are potential suitable sites on the geography for this species. So that's just to have the two concepts that are very useful here: ecological niche and geographic distribution. So now I'm going to mention which are the two species of concern and that we're modeling in this exercise. Big concern for forest ecosystem in Mexico. These are ambrosia beetles of concern. Ambrosia beetles are wood-boring beetles that feed on fungi, and they have associated bacteria. So in this picture that I took from the ambrosiasymbiosis.org webpage, where there is great information on these beetle species and their fungi they feed on, you can see it's a real picture on how these beetles carve into the wood of a tree. And then you have the larvae of these beetles, and then the fungus where they feed on, right? So the beetles have on their mycangia the fungus that when they drag into the tree, they can deposit, and then using the tree vascular system, this fungus reproduces. And when the beetle starts reproducing inside the tree, they feed on this fungus. So what ends up killing the tree is the fungus. There is chemical signaling on which tree species these beetles can infect more frequently. So there is semiochemicals that are more abundant in decaying trees or stressed trees. This is the cartoon on the right. Imagine this is at least what happens in pine forests. And so a healthy tree that is on the left has a less number of beetles than a decaying tree that is on the right. And as I'll show later, these trees, well, I'll talk about that later. But then you have a female that comes out from this tree and is the one that can fly and arrive to a new tree and infect it. There are chemicals that the beetles can follow and track to know which trees to infect. And the way this works is basically when you have a healthy tree, there is a higher concentration of terpenoids that is much less on a dead tree or a decaying tree, basically a stressed tree. And the concentration of ethanol from a healthy tree to a dead tree increases. There is a whole gradient of beetles that can, or diversity of beetles that can infect a tree depending on which stage of, or how healthy a tree is, basically. Ambrosia beetles are usually attacking or what are stressed trees. And so I emphasize on this because we have to then think on what environmental predictors are going to be more useful for predicting the distribution, potential distribution of these ambrosia beetles. So fair enough to say that there is a chemical recognition and that we can think on healthy or stressed trees for modeling the distribution of these beetles in Mexico. The two species of concern that we are talking about here are these one, Xyloborus glabratus, both come from Asia, and Wallacea fornicatus, that not only from Asia but also from Africa. Xyloborus glabratus arrived on the east coast of the United States in 2002. What is embalaje? Well, through merchandise on boats. No, these are very, very tiny beetles, around approximately 2 millimeters, so really tiny, difficult to detect for any office that is in charge of taking care of this in the ports of entry. And the native hosts are several families of species, basically Lauraceae species. In Asia, this is not a problem. No, there has been a long path of coevolution between the beetles and the trees there. But in the Americas, these species are exotic and have been showing a lot of pathogenic problem in native and cultivated forests. This species, Silebros glabratus, carries a fungus called Rafaelea lauricola. It's its symbiont, and this is a fungus that it feeds on, and that eventually causes the death of the trees, as I showed before how it works. The other species is Eulachia fornicatus. It causes it's a polyphagous shot hole border. That's how it's called also. Hosts in its native range correspond to this family, Thiaceae, but also can infect more than 50 other tropical families. It was introduced in the west coast of the United States, in California, in 2003. Was observed there in one tree species, and since 2012 it has been spreading with like alarming rates, not only in urban ecosystems but also in native ecosystems, like in forest ecosystems in the west coast of the United States. There is a new report that has shown 335 tree species that have been affected by this beetle, and 207 with the beetle correspond to 50 that have symptoms and signs of the infection, correspond to 58 families. So the host range of this beetle is quite large, and consequently very important for not only crops but also native forests. It transmits 3 fungi, and so yeah, larger risk for infection of other trees. So the question here is, what is the potential of exotic beetles to infect or interact with new hosts and beetles? So the transmission of the disease or could be where, and yeah, can we assess that? How bad could it be for other forests? These are some pictures. This is from this report from Akif Skalen in 2013, Plant Disease. And this is only for the Eulachia species. You can see on the upper left panel, letter A is the female of this beetle, and the letter B in the middle corresponds to the male. And look at the size of these beetles. This is a penny. And you can see these tiny dots here. This is the female and the male. So these are really, really tiny beetles. Almost flightless, but so tiny they may be able to move easily through other forces. These are pictures in the bottom panel, ABC, that correspond to the symptoms, external symptoms in 3 plant species. The first of it is the avocado tree, Persea americana, on the panel A, and other species like Acer and Ricinus. So this, you can see the penetration of beetles where it has happened, and it starts causing problem inside the tree, which you see now. You can see the galleries that the beetles start digging after they infect or start digging into the wood. different ways of infection here, all the galleries where they reproduce, and also the fungus that they carry. And the fungus starts spreading through the vascular system and finishes killing the tree. That's what happens with these pests. So now I'll show you the diversity pattern of Lauraceae species worldwide. And the idea of doing this exercise at a global scale is to look at where could we see potential risk for the infection of these beetles, and also where could we look at interesting questions from an evolutionary perspective or a pest control if we wanted to do that exercise, right? So looking at the diversity of these plants worldwide can be practical for these studies, not only from a modeling perspective, but more for the people that has to go there and sample and look at the system very closely and take samples to the lab and start working for pest control. So for this first part, I used GBIF records. This is a compilation of 1,241 records in the grid. The grid that I use is a 2x2 degree, and 400. Oh, this is a mess. Yeah, 406 species, and then I have a mess here. 2,564 species. Okay, I'll show you. And then all these analyses were done in ArcGIS and R. No? But finally, the degree is a coarse resolution degree, grid, I won't say, because this is based on occurrence data, not distributional maps, and I thought it was fair enough to do it coarsely. So where are the Lauraceae species? These are, I confirm here, it's 406 species that I compiled from the Global Biodiversity Information Facility and what was available through the National Commission on Biodiversity from Mexico. You can see on the map in the center, the big map, is where are all the occurrences of Lauraceae species that are at least present in these databases. And on the lower right panel is what the angiosperm phylogeny group has as where the Lauraceae species may be present, right? So the correspondence of the maps is good between these two. However, the quality of GBIF data may be lacking because we know not all the species have been sampled, or not all the herbaria have given their data. Nevertheless, this is important, good information that we can use to start looking at patterns here. Lauraceae richness. So this is a map I'm showing here, and we will start talking about the two views that I mentioned before. Remember, on the range diversity plots, we have two views. We have the views by sites or by species. In this case, we're looking at by sites. So we have sites that have from 1 species to 206 approximately. Some hotspots of diversity appear in East Asia, Oceania, Madagascar, parts of Africa, but most of the records that we have are present in the Americas and mainly the Neotropics, Central America, Northern South America. So the big concern for those beetle species to attack these forests, given that they are native from Asia and they don't cost much trouble, but if they arrive to the Americas, it could be very drastic, probably what the consequences are if they arrive here. If we look at the dispersion field, remember, is on average how widespread are the ranges of the species that inhabit one cell. So you see this is a different scale on the lower left panel. Red corresponds to sites that on average contain very widespread species of Lauraceae, and blue would correspond to very narrowly distributed species on average. So you see there are parts of, let's say, Australia, in the north part of Australia, that we see that there are Lauraceae species that are very widespread there. Africa apparently contains or shares many of these species with other parts of the world, and other parts here, for example, in islands and things like that. So those species, most of them are probably some cultivated trees. And because we don't have a right now very clear view on what species are these, but some of the very widespread species for sure are cultivated. Anyway, think about this on how can we look at the risk of invasion of these beetle species, say if on average the species are very widely distributed or narrowly distributed. So as I mentioned before, hotspots of species richness are now highlighted in these black cells. Those would be sites where coevolution may be interesting to study in the east part of the world, in Asia, or where there is probably higher risk of losing a lot of native Lauraceae or also cultivated Lauraceae in the Americas. Now, if we look at the lower species richness, we're looking at those sites there. And okay, this is okay. So this, as I'm highlighting here in the lower right panel, you see I'm highlighting those sites that are very rich, species rich. This is the red square that I'm using here. So remember, the y-axis in the range diversity plots corresponds to species richness. And so these sites are, on average, narrowly distributed, but very rich in species. So those species are important because not only these are species-rich sites, they are also narrowly distributed sites. Now, if we change our view to this slide that I'm showing here, now I'm highlighting on black cells what corresponds to very widespread species on average that are in general very species poor. But so these sites may be important because these contain species that are widely distributed, and the potential of beetles to arrive there and infect and spreading out from those systems to other parts, other ecosystems, forest ecosystems, may be very important. Now, this is the view by species of some of the Lauraceae species that are selected. These are the 5 species that are with larger geographic ranges. Remember, this is the view by species. So now on the y-axis we have proportional range size, and I'm using this red cell to highlight which species I'm selecting. You are looking at the lower right panel. And on average, these species share their distribution with between narrowly few species or a lot of species. Not so many species, but quite a bit when you move to the right on the x-axis. So which are these species? We know of one, which is avocado trees, Persea americana, that's in green, dark green. And you can see that in fact Persea americana is widely distributed. Some other species are these that I have here, the Casita filiformis, Laurus nobilis, Nectandra cuspidata, and Ocotea cernula. So these are species that are with large range and low diversity. Now, if we look at the community of beetles, this is the slide that I'm presenting here, we see there is a huge sampling bias. We don't really know much when we use GBIF information on where the beetle species relate that correspond to this genus, Xyloborus, are. We know they are very abundant in Asia, but we have very few information here. And so we have to start using other databases rather than GBIF, and that's something we're doing to complete this exercise and have a better representation of their distribution and later niche models. But so look at the, well, fair enough, let's see, there are sampling biases. Some Northern European countries have a lot of information there, and then scattered occurrences in the Americas. And when we include other local databases like the CONACyT, we see our sampling starts improving. So that's something we have to have in mind, and makes sense to mention it here in this global biodiversity seminar. We have to share data and start improving our databases. And, well, the species of concern is now here represented by green dots. And I mentioned before, this is a species that arrived to the East Coast in Georgia in 2002 and has been moving westwards because of reasons I'll mention before, but basically humans have been distributing, spreading it because they sell the wood for barbecue and things, yeah, wood for fire. So let's take a look to the distributional patterns of this species in Mexico. Again, I'm using, well, now I'm using niche models to predict the potential distribution of both the Lauraceae species and Xyloborus species, and, well, what we call supervised niche modeling, which is basically taking care of well, sampling biases, spatially and environmentally. We are building what we call the M in the BAM diagram. You've seen seminars of this before, but the idea is that you have to train the ecological niche model in a region that would correspond to what has been accessible in a relevant period of time to the species. So it's not ideal to train a model in the entire world, let's say, but to restrict it to where we can hypothesize has been the accessible area for the species, and that's what we call the M in the BAM diagram. Something I'm sure you've seen in other seminars of this series. We then have built two presence-absence matrices to show the diversity patterns after doing all the niche models for the species. So we have 89 species of Lauraceae, and because we're using a higher resolution grid, which is approximately 5 by 5 kilometer, we have many more sites. We have above 317,000 sites, right? So the presence-absence matrix increases its dimension just because we're increasing the resolution, spatial resolution. Then for Xyloborus species, we were able to collect 20 information of better quality for only 23 species that are distributed in the Americas, and the number of sites at this spatial resolution is approximately this, no? It's 224,000 sites. Again, all this was done in ArcGIS and R. So let's take a look to Lauraceae species in North America. A little bit of Central and South America, actually. We have on the left panel, the map on the left, what corresponds to the species richness of Lauraceae. So remember, I'm using the range diversity plot to point at these two measures of diversity. The left panel, species richness, the right panel, the dispersion field. So let's look at the left panel first. This would correspond to the y-axis in our plot, range diversity plot. So you see most of the species diversity of Lauraceae is concentrated in the highlands in the higher parts of Mexico going south, and then towards Central America. And then you can see a few other yellow areas in Mexico that correspond to other sites of species richness. This is pretty much everywhere in Mexico except for Baja California, the very dry parts in Sonora, and northern Mexico close to the border. The rest of the territory in Mexico going south towards South America is where we find the hotspots of species richness of this plant family. If we look at how widespread are those species inhabiting those sites, we have that in the right panel here. That's what we call the dispersion field. So in blue, you have areas where you would expect to find very narrowly distributed species on average. And then on red, you have what would correspond to very widespread geographic ranges. So you can see there are a few species very widely distributed from the east coast of the US, part of the west coast of the US, and many parts in Mexico, right? And so that's what would be very important to consider for monitoring the spread of the beetles and the fungi that they carry, because this can be the favorable sites for the species to establish and continue spreading and causing problem in these forest ecosystems. If now we're looking at the Xyloborus genus in North America, Central America, we see the same pattern. We have that the highest concentration of species of this genus of beetles is go towards the Gulf Coast in Mexico, and then increases towards Central America. This is in reddish, yellowish colors. Some species of these beetles are not so rich in the east coast of the United States or other parts of the continent. No? But see the correspondence between this map of the species of Lauraceae and this map of the distribution of Xyloborus in the Americas, and they match very well. And that the reason of that is also because they have coexisted for a long time. Those are the types of forests where you find these species. Then, if we look at how widespread are these beetle distributions, you see the same pattern. Well, not the same pattern, but I want to say is in the sense of using the range diversity plots, is some sites have species that are narrowly distributed in blue, and in red, those species that are very widespread. So I guess this is important to think on when we want to go to the field and start thinking on experiments to do sampling, and basically also advising authorities that have to take care of cultivated and native forests, urban areas, or natural areas, right? Let's talk about briefly on the potential distribution of the Xyloborus glabratus. This is an exercise using spatial multicriteria evaluation. So I'm using several maps, suitability and distribution of the species of Xyloborus that we're interested, which is glabratus. And then we have the suitability of all the species where these species coming from Asia could be in contact with that are naturally distributed in the Americas. And also, what are the plant species that may be affected, in this case only Lauraceae species. We are using niche models based on NDVI, which is a vegetation index from remote sensing sources. And also we're using stress factors related to plagues and infrastructure and fragmentation and number of dead trees. And for this exercise, we're using all the niche modeling tools that we have, I don't know, we commonly use, but also ILWIS, which is an open source software where you can use the spatial multicriteria evaluation tools. So this is what you have to do to the spatial multicriteria. You have to define a decision tree. Our target is to predict or have a surface related to the suitability of the beetle species Xyloborus glabratus in Mexico, and we are relating it to two to three factors that we look at in two different levels. So first, where is the potential distribution of the beetle, and where is the favorable conditions for infection given the distribution of trees and other Xyloborus species that may be able to carry the fungus, the Raffaella lauricola, that this beetle has. And finally, where are forests that may be more stressed? You have to give weight for these layers, and the weights are given in parentheses, 0.4, 0.35, 0.25. We have several layers for each of these set of factors. So for this distribution of Xyloborus glabratus, we have the median of the suitability and the thresholded map. So the median of the suitability is a map that contains continuous, is a continuous variable. But we know that not all the niche model that comes from that map is a, let's say, we are using Maxent, and Maxent has to be, output has to be first like thresholded to have an idea of the presence, absence of the potential distribution of favorable conditions. And in the median, when we use a continuous variable using the median suitability, we know that low values are very abundant and that high suitable values are not so abundant. So what we do here is we standardize the variables according to what we think is more convenient in each case. For the distribution of Laraceae and other Xyloborus species, we have the same idea. We have where is the suitable condition for all Laraceae species all together, and the thresholded map for this niche model, and the same for where are the suitable conditions for all Xyloborus species in Mexico, and the thresholded map. Finally, we have three other factors that we thought may be important to include to have a better prediction on where could this beetle be more dangerous. And this is what we mentioned before, stress in forest ecosystems. So the probability of finding plagues on trees, the probability of finding dead trees, and a variable that corresponds here in MexBio is fragmentation and infrastructure. So like where are those ecosystems that are not necessarily under stress, but maybe more related to stress factors. So if we look at the first part, what we call the Xyloborus glabratus potential distribution, we have the two maps that we mentioned. We have the median of the suitability in the upper right panel, and the thresholded map corresponding to this suitability in the lower right panel. The way we predicted this suitability map in Mexico comes from projecting the model trained in the eastern coast of the United States using NDVI into the Mexican territory. We use the five principal components of NDVI from the period of time that is here, 2002 to 2014. Then you have what corresponds only here, I'm not showing the thresholded maps, only the suitability of all Laraceae species in Mexico and the suitability of all Xyloborus species in Mexico. So remember, if the species of concern, Xyloborus glabratus, gets in contact with native beetles that are related, the chances of transmitting the fungus increases, and so that's why we thought it was important to use all the Xyloborus species as well. Finally, we have the third set of factors that are related to stress, and you see in darker red in all the maps, I omitted to say that, but you have darker corresponds to higher probability in the upper panel, what corresponds to tree plagues. The middle panel corresponds to dead trees, and the lower panel is that variable that corresponds to infrastructure and fragmentation in ecosystems in Mexico in general. It's a very complex index, but I'm just simplifying it, as I said here. So if we combine all these using this decision tree, we get this prediction for Xyloborus glabratus in Mexico. And so if we were to advise Mexican authorities, or we had to go and look at, well, where to put people and in the ports of entry or other regions for this beetle species in particular, you can see in red where we would expect, according to the values of the variables that we have used, the higher risk of invasion of these beetle species. So I have to mention, and you have to be thinking that you can change the weights of this decision tree that you see here. You can increase or reduce the number of factors, the number of levels, according to different criteria. And so you shouldn't think of this as something very rigid. Instead, you can think on this as a tool that we can improve, we can change, and that we can use according to our needs and what we think may be more important for predicting the potential of these plagues in Mexico. So finally, because Wallacea species has demonstrated to be a very dangerous beetle after its arrival to the West Coast in the United States, would be interesting to start taking a look to the distribution of some species that have been affected by this beetle species and the fungus it carries, and if Mexico may be under risk or not, how far we would expect that to happen if it's not there already in northern Baja California. So we have compiled the distribution of the few species that we know are reproductive hosts of this beetle. These are one of them, the ones are here: Acer negundo, Platanus racemosa, Populus fremonti, Populus trichocarpa, Ricinus communis, Salix godingi, Salix levigata. So you see, they are distributed the West Coast, touching the border with Mexico. Some are going down through the parts of the Sonoran Desert. Those are the occurrence data that we have. And if we apply tools, niche modeling tools, we can have probably a better prediction on potential distribution of these beetle species. I'm presenting here only the suitability maps. I haven't shown you the thresholded maps. I don't have them with me in this presentation. But you see where the clouds of points for each of these species are in black dots, and the suitability of these species. And so you can see if the beetle has arrived to the West Coast, in Los Angeles, in Riverside, where it has shown tremendous, well, terrible impact in forest ecosystems, Mexico, through Baja California, parts of the northern desertic ecosystems, shows also a lot of potential for spread. So other things to consider are how can these beetles spread more faster, or how can they move farther, because they are flightless, but how can they be more in danger of affecting other ecosystems? So in the case of Xyloborus glabratus in the East Coast, we've seen huge jumps in the distribution, the spread of the beetle going west because of people selling wood for fire, and like barbecue and things like that. So since its arrival in 2002 in Georgia to the present, 2014, the beetle has been moving and jumping very far, and that's because mainly human causes. Other things that we may think is agricultural imports. These beetles have arrived to the Americas because of transportation of I don't know, goods to the Americas coming from Asia. So we have to look carefully in the ports of entry. There should be people in charge of taking care of what comes from Asia that could be of risk for having these beetles. Not only then directly from Asia to Mexico, but also from the US into Mexican territory. And so you see there are several ports of entry where merchandise comes to the country. And the other agent that can be of importance is storms and hurricanes, for example. So what I'm showing here in this map in the background is all the routes of storms since 1850 to 2013. And so these are the blue lines that you see there, right? So we could also have, that I haven't shown you here, but a map of where are the mean routes of storms into Mexican territory that could potentially help spreading these beetles if we think of them as strong enough to carry them. I mean, think of this of thinking about what other agents can be important. I've mentioned before the need to share data, and so basically people that is in the field can contribute a lot. Experts in the taxonomic groups have helped with giving us databases that are very of much quality from what we get online. There may be routes that are already logical to do and act to advertise people in the field and producers. For example, there is colleagues in Riverside that have already experienced the problem of having the Wallace species and the problems it has caused. We can use their information to update mobile devices so that we can use faster to get pictures how are the signs and symptoms of these trees after they are infected or by this beetle species, and that we can eventually use to share with Mexican authorities and spread the word at the governmental level, and not only the governmental level, but also with everybody, right? The entire community. So, for example, this is a map of botanical gardens in Riverside. The botanical garden there has had a huge, drastic impact from the invasion of this beetle. So, one way to spread the word would be to talk to the people in charge of botanical gardens in Mexico, in the US, increase that community to schools and other communities where we can not only tell them to tell us if they know, but also use them to retrieve information that may be important. Finally, I just want to quickly mention the important role of citizen science and field technicians. So remember, this talk has been inspired, and the information that I've used was also based on what people in the field technicians have experienced, and they know it's important for the spread of these beetle species. So we can use them to give have a close contact and exchange ideas how this can be improved in the quality of our response. And important role of citizen science, meaning that we can retrieve information from sources that are online, and we can, as I mentioned before, update web pages and have people have access to online resources where we can look at the signs and symptoms of these diseases, right? So this is important to act faster. Other tools, what I have presented, are already have been developed in collaborative efforts at the University of Kansas. This is something I would like you to try if you like GIS and like the idea of trying to start measuring things like the range diversity plots. There is a Python plugin already available for QGIS that was developed by the Biodiversity Institute in collaboration with the Informatics people and Jorge Soberon and other colleagues from the Biodiversity Institute there that already measures all the range diversity plots, and it's incorporating the phylogenetic part as well. So this is the idea. You have that plugin for Quantum GIS. This is an open source workflow and web service that you can use to do these measures. Well, that's all that I probably have to say for now. I hope it was clear. If you have questions, please let me know. I know I sometimes I not speak very clearly or have forgotten to mention something that was key. But I hope the idea was clear, and you can feel free to contact me if you have questions. This is something that is ongoing. It's a recent analysis, and hopefully we can finish it soon to have a manuscript of all this. Thank you.
Thank you, Andres. That was great. If people have questions, as always, we have this thing of, do we wait around for questions to come in? And they always do, but it's rather a bit uncomfortable in that we also don't want to keep you all online very long. This is the email to which you can send your questions. If you have questions for Andres, you're very, very welcome to email to this address or to post on the Google Plus page. And I'm sure that Andres will be willing to respond to you. I haven't seen any questions come in yet. So I think we'll end the broadcast here, and again, give us your questions as soon as you can, and we'll share them around. Oh, actually, here's a question from Monica Papes. She's asking you, what is the source of your data on storms and hurricanes?
This is from online, Mona. Is the NOAA? It's the NOAA. So yeah, I have it. It's available online. It's free, and there is all the tracks of those storms, and I can give it to you if you want, for the entire world.
Okay. Thank you very much, Andres, and everybody else. Thank you. If you're in the U.S., thank you for appearing on Thanksgiving morning. And if you're not in the U.S., we will announce a seminar for late January. So thanks again, and goodbye, everybody.